Go ahead, prorector.
Yeah. Okay. Welcome, everybody. Welcome to the candidate. Good to see you. We will now start with a ceremony. And I open this academic ceremony in which Huajie Liu will defend the academic thesis, Deep Brain Stimulation and Memory Restoration. May I invite you to present a summary of your study and the conclusions of your thesis.
Yes. Thank you, dear prorector, dear members of Corona team, dear family, friends, and dear colleagues. Thank you for your attending the defense of my PhD thesis, which is entitled Deep Brain Stimulation and Memory Restoration. In the next 15 minutes, I will present a brief summary of my thesis. Dementia is a general term for a group of brain disease, which is characterized by the degradation of neurological, behavioral, emotional, and psychological cognition and function. There are different types of dementia, such as Alzheimer's disease, vascular dementia, Lewy body dementia, frontotemporal dementia. The most prevalent cause of dementia is Alzheimer's disease, which is the cause of 60 to 70% of cases. A new case of dementia occurs every 4 seconds around the world. According to statistics, the prevalence of dementia among people over 65 years old is high as 7% around the world. The higher prevalence of dementia in developed countries may be due to longer life expectancy. There are almost 47 million people have dementia by 2015, and this number will be expected to increase to 132 million by 2050. The most common early symptom is memory loss. As the disease progresses, symptoms may include disorientation to time, difficulty with day-to-day tasks, such as cooking or shopping, hallucinations and paranoia, decreased social interaction and withdrawal, repetitive questioning, difficulty speaking and writing, confusion and disorientation. Pharmacological treatments are not effective for every AD patient and only improve symptoms temporarily. Moreover, there are still some substantial side effects, such as gastrointestinal symptoms, eating disorder, and weight loss after using drugs. So, urgent need for alternative treatment options. Recently, researchers found that deep brain stimulation could restore memory loss. It may regulate neural activities in memory circuit of the brain. DBS is a neurosurgical procedure in which stimulation electrodes are stereotactically implanted into specific brain target and local or general anesthesia. The DBS system consists of 3 components: the implanted pulse generator, the electrodes, and an extension. The stimulation parameters can be adjusted externally with a wireless connected controller to obtain best possible therapeutic effects with no or least side effects. Some important features of DBS therapy as non-ablative and reversible nature, as well as the adjustability of stimulation parameters to the need of individual patient. Traditionally used to treatment movement disorders, such as Parkinson's disease, tremor, and dystonia. Recently, studies have begun to explore the applicability of DBS in a wide array of conditions, such as dementia. Thus far, stimulation targets in cognitive disorders were selected within the circuit of puppies and related limbic structures. The phonics stimulation can lead to decreased rates of cognitive decline, enhanced visual spatial memory, and improving verbal recollection. While the exact mechanisms of action are not completely understood, studies suggest phonics DBS to be involved with increased functional connectivity and neurotransmitter levels, as well as enhanced neuroplasticity. A decline in the number, size, or function of cholinergic neurons of the nuclear basalis of minute may be associated with impaired cognition and memory in dementia. It has been shown that NBM-DBS can influence activity in pathological neural circuits that underlie AD, although optimal stimulation parameters and mechanisms of action are yet to be elucidated. Since non-surgical lesions restricted to the circuit of puppies are rare in humans, the study of memory has greatly benefited from animal experiments. Lesions in the puppy circuit have been found to have a profound effect on learning and memory, whereas spatial and non-spatial memory performance. In our group's previous research, we found bilateral phonics DBS could improve spatial memory performance in the object location task and in a rat model of experimental dementia. And we also found phonics DBS could enhance acetylcholine levels in the hippocampus. Researchers have found that DBS may regulate neural activities in memory circuit of the brain, which has encouraged the initiation of dementia-related research. It has been shown that DBS of the nuclear basalis of minute can influence activity in pathological or neural circuit that underlie AD, although optimal stimulation parameters and mechanisms of action are yet to be elucidated. In this research, we used bilateral electrodes in the nuclear basalis of minute. Rats were divided into two groups, one sham group with stimulation cable, DBS off, two DBS group with different parameters. Rats were tested in the object location task with the following conditions: one, low and high frequency, 20 Hz or 120 Hz, two, monophasic or biphasic pulse shape, three, continuous or intermittent DBS, 20 seconds on, 40 seconds off, and 100 microampere amplitude. Thereafter, rats were stimulated with the most effective parameter, followed by BRDU administration and perfused 4 weeks later. We then evaluate the effects of NBM-DBS on hippocampal neurogenesis, synaptic plasticity, and on cholinergic fibers in the perirenal and cingulate cortex using immunohistochemistry. We also performed in vivo microdialysis to assess circuit-wide effect of NBM-DBS on hippocampal acetylcholine levels during on and off stimulation. The bilateral electrodes were all implanted within the histological boundaries of the NBM. We employed slightly different stimulation parameters and found that beneficial stimulation parameters entailed 20 Hz stimulation, 100 microampere amplitude, as well as biphasic pulse shapes. Using the most optimal stimulation parameters derived from the OLT, we found enhanced recognition memory performance of NBM-DBS rats when compared to sham. In the open field test and elevated zero miss, no clear evidence for anxiety-related side effects after DBS was found. The immunohistochemical CFOS results revealed evidence of enhanced neural activity in the entorhinal and perirenal cortex. The entorhinal cortex plays a significant role in spatial memory, while the perirenal cortex is involved in recognition memory. This is in line with our findings of improved memory performance in both the ORT and ORT following NBM-DBS. Additionally, we also found enhanced neural activity in the three main hippocampal fields, C1, C3, and dentate gyrus, which all play an important role in spatial memory. We also found that acute stimulation promoted proliferation in the dentate gyrus, increased synaptic plasticity in the C1 and C3 subregion of the hippocampus, and increased the length of cholinergic fibers in the cingulate gyrus. However, there was no difference regarding hippocampal acetylcholine levels between the two groups. These findings suggest that the potential mechanisms of action of the induced memory enhancement through NBM-DBS might be due to selective neuroplastic and neurochemical changes. Then we evaluate the effect of intermittent and continuous STN-DBS on cognitive functions. A total of 10 patients with PD were randomly assigned into two different DBS treatment groups. Group A was treated with intermittent STN-DBS, and Group B underwent a continuous STN-DBS. The motor and cognitive assessment, the simple reaction time, and the complex reaction time performance were all assessed before and after the DBS surgery. In addition, the lumbar punctures were conducted 1 day before and after DBS surgery to obtain CSF samples and measure neurotransmitter levels of acetylcholine and glutamate. Results show that intermittent STN-DBS seems to be more effective in terms of cognitive function when compared to continuous DBS. Furthermore, the increase in CSF acetylcholine levels may be linked to the cognitive effects of intermittent STN-DBS. So, summary, performing DBS of the phonics or in the NBM seems to be a safe, well-tolerated therapy without causing new neurological deficits or permanent stimulation-induced side effects. The NBM-DBS with optimal stimulation parameters has the potential to improve memory function in conditions characterized by memory impairment. Additionally, there seems to be a causal relationship between stimulation-induced promotion of adult neurogenesis and spatial memory enhancement. The intermittent stimulation model of DBS also seems to be an effective and safe treatment for cognitive function in PD patients, albeit in a different brain target, such as the subthalamic nucleus. Ongoing clinical trials will need to demonstrate that DBS can lead to a reduction in the clinical significance of disease progression, so that DBS can be used as an established treatment option for patients with dementia. Thank you for your attention. I'll give the floor back to the prorector.
Thank you very much, Mr. Liu, for your presentation, very clear presentation. We will now start with the opposition, and the opposition will be opened by Professor Zhang Zhen. He's neurosurgeon at Shandong Provincial Hospital in China. Welcome, Professor Zhang Zhen. The floor is yours. Please unmute yourself. Unmute yourself, please. Professor Zhang Zhen? Can you unmute yourself? He can't hear you. Yeah, okay.
Okay. And I'm Dr. Shantai Xu from Shandong Provincial Hospital affiliated to Shandong University. I have two questions with Dr. Liu in his thesis, chapter 4. And in the NBM stimulation, you set up 8 different stimulation patterns. And eventually, you found just one pattern was effective, and the so-called intermittent biophysic stimulation. So here, different stimulation pattern seems to have a different effect. And what's the mechanism behind that the different stimulation have different effect from a neurological point? And the second question is about the dialysis experiment. In the dialysis experiment, you just include 2 groups, and the NBM group and the DBS group and the sham group. But you were doing dementia experiment, dementia research, but you didn't include the group, dementia group. Why? Just 2 questions.
Yeah, highly esteemed opponent, thank you for your questions. I will first answer the first question. And you asked me the different mechanisms of the different stimulation parameters. And the effect of the NBM DBS depends more on frequency than current density. Probably because the NBM neurons typically have a low frequency firing rate, and the low frequency stimulation is believed to activate the cell bodies. And high frequency continuous stimulation is used for movement disorder as a way of inhibiting their output. And the high frequency stimulation, usually about 100 Hertz, and was successfully used to stimulate the STN and the globus pallidus internus in patients with PD patients. However, no overexcitation has been hypothesized in Alzheimer's disease, and lower stimulation frequency or intermittent patterns of stimulation are likely to be more effective for stimulation of cholinergic fibrin. And in our group's previous research, the phonics DBS could enhance memory functions in experimental dementia. And then we found that the phonics is not sensitive to the frequency of stimulation, but rather to the current levels, since both 10 Hertz and 100 Hertz were all efficient. And however, in the case of 10 Hertz, we needed higher currents to reach therapeutic effects than when 100 Hertz was applied. This finding suggests a total current density threshold rather than a frequency effect. And the frequency well known for DBS of the STN in patients with PD, as well as corresponding animal models, where only higher frequency produce therapeutic effects. I think the major difference is that in the Parkinson's disease and in most of other neurological disorders, the gray matter is usually stimulated to carry overall inhibition, although some excitation also occurred. And then furthermore, in some DBS implanted epilepsy patients, as well as in animal models. The high frequency in combination with high current stimulation of the anterior thalamic nucleus impairs rather than facilitates cognitive function due to an overall inhibition of the limbic structures. This is the first answer of your question. And I will, the second question you asked me, why not use the scopolamine-induced dementia rat in the microdialysis? And then I think scopolamine is usually used as a standard drug for inducing cognitive deficits in healthy humans and animals. For our research, we use a scopolamine-induced model to investigate which stimulation parameters to use and which is the optimal stimulation parameter. It has been shown that the anticholinergic action of scopolamine more readily impact the hippocampus than other structures. Previously, we have found that the phonics DBS could increase hippocampal acetylcholine levels. However, in the microdialysis, we would like to assess circuit-wide effects of NBM DBS on hippocampal acetylcholine levels during on and off stimulation. For our research, we first wanted to check what stimulation does in a healthy condition, and we tested it in the hippocampus. However, we found no effect. And for the next step, I think we will then use the microdialysis in the frontal temporal cortex, and we will investigate in the scopolamine model. And I think we first want to check the healthy conditions, and then we will also investigate it in the model of disease. That's my answer for the two questions. Do you satisfy with my question, my answers?
Okay, thank you very much.
Okay.
Thank you, Professor Xiangxian. We would like to continue with the next opponent, Professor Blokland. Professor Blokland is Professor of Comparative Neuroscience at Maastricht University. Professor Blokland. Please unmute yourself.
Thank you, Pro-Rector. Dear candidates, first of all, I would like to congratulate you on your thesis, and of course also your team that supported you. What I hear is that in the last year from a long distance, but I still managed to finalize the project with you. That's really nice. I read the thesis with great interest because I also was involved in this work, and although there's a lot of work that shows that the brain stimulation could be potential treatment for dementia, there's still work needed to really understand how this works and how it can be really beneficial for patients. So, and I hope this work will continue and maybe will lead to a treatment in the future. And I think your work contributed to that. So, good work. But of course, we have still a lot of questions in this field, and we also would like to get a better understanding of what DBS is actually doing and how we can improve this procedure. So I would like to go to chapter 5, because that's of course the ultimate challenge. Where we would like to see how patients can benefit from this treatment. What I was a little bit surprised by the method that was used in chapter 5, the intermittent and continuous stimulation. you already explained a little bit about the differences in the intermittent and continuous stimulation parameters with the previous question. But the first clinical question is, so if you have 20 seconds off and 40 seconds, so 20 seconds on, 40 seconds off intermittently with these patients, what I remember from, I've never seen a real patient, well, yeah, I've seen patients, but only with stimulation on. But when I look at YouTube movies, what you see an instantaneously change of the clinical picture when you turn off the stimulator. So with the patients that you had, you had 20 seconds on and then 40 seconds off. What I would expect is that during the 40 seconds off, these patients would go back in a state of Parkinson's disease, but they would start trembling, showing tremor, and all the Parkinson-related clinical features. Maybe that's the first question that I would like to ask you. So what was the clinical effect of the intermittent stimulation? We have 40 seconds off.
Dear highly esteemed opponent, thank you for your kind words and thank you for your question. You mentioned that I use intermittent stimulation, 20 seconds on, 40 seconds off. You would like to ask what's clinical effects of the intermittent stimulation when we have 40 seconds off, during the 40 seconds off. First, we used this intermittent stimulation just after 1 day after the surgery, and most of the patients have good conditions just after the surgery, and I think 20 seconds on, 40 seconds off is better for their motor syndrome. In my research, the patients, most of the patients have good conditions during 20 seconds on and 40 seconds off stimulation.
So you don't see any Parkinson-related symptoms during the 40 seconds off?
You mean I didn't see any Parkinson-related symptoms as the pre-surgery, such as the pre-surgery?
Yeah, exactly. Yeah.
I think most of the patients did good after the stimulation. I think in the motor syndrome, the intermittent stimulation did as well as the continuous stimulation.
Yeah, I still don't understand. So it seems that intermittent stimulation. So when you have 40 seconds of no stimulation, so from what I know from DBS in Parkinson patient, if you switch off the stimulation after continuous stimulation, you see immediate clinical effects coming back again.
Yeah, and some studies have also shown that short high frequency stimulation of STN helps to increase partial discharge frequency, and then it is inhibited with the extension of stimulation time. And another study also showed that the STN DBS would interfere with the ability of a subject to appropriate slow down their response when betting on their preference, result in a reduction in response time. And the intermittent DBS enhanced this. In a basic neurophysiological study of using intermittent DBS instead of continuous DBS in patients with epilepsy, intermittent DBS can also lead to the reduction of potential cumulative evoked potential lasting longer. I think this suggests the existence of homeostatic plasticity. And another study also showed that the axon afferent flow can be activated by each intermittent stimulation pulse. Thereafter, the intermittent DBS could activate STN axons at each pulse transmission.
Okay. Okay, maybe a short next question related to the neuronal network of the basal ganglia and stimulation of the subthalamic nucleus. So you observed an increased level of acetylcholine in these patients after intermittent stimulation. Via which network? I mean, when you look at the network in the basal ganglia, how could you expect an increase in acetylcholine when you stimulate the STN? So it's going to the thalamus and then the cortex, and it comes from the other regions of the cortex nucleus. So how could you explain the increase in acetylcholine based on the neuronal network of the basal ganglia?
You mean you want me to explain the increase in acetylcholine based on the neural network of the basal ganglia?
Yeah.
Yeah. And I think the hippocampal acetylcholine levels were not affected by NBM-DBS, since there's no direct hippocampal cholinergic input from the NBM. However, it has been claimed that the anticholinergic action of scopolamine may more readily impair the hippocampus than other structures.
No, I mean the STN, so the stimulation of the STN.
You mean during the STN stimulation?
Yeah.
Okay. I think although it is not clear which neuron components are the source of acetylcholine release, and I think the possible explanation for the result is that intermittent STN DBS may activate the cholinergic system and induce acetylcholine release. And several studies showed that the cholinergic neurons in pendaculum pontine tegmental nuclei project to the STN. And furthermore, the pendacular pontine nuclear DBS, the PPN DBS also showed better clinical outcomes when combined with STN DBS. I'm not sure the exact mechanisms of the STN DBS affect the acetylcholine, but I speculate it may be in this pathway.
Okay,
thank
you. I give the word back to the rector.
Thank you very much, Professor Blokland. The opposition will be continued by Dr. Lim. Dr. Lim is assistant professor at the School of Biomedical Sciences at the University of Hong Kong in China. Professor Lim, welcome. The floor is yours.
Thank you, Pro-Rector. First of all, congratulations to supervisors and to the successful of your thesis, Huajie Liu. So as we know, my first question is, as we know that the molecular mechanism for C-force and synaptophysin inductions, which takes several hours to several weeks, and also the process of neurogenesis from several hours also to days. Okay, for example, from an immature neuron and to become a mature neuron, it takes 4 weeks. So how do we know? My first question is for your chapter 4. How do you know that the improvement of memory by NBM-DBS is due to the increase of C-force and also the increase of your BRDU or new N and also the synapse? because I think your stimulation is spontaneous and fast.
Dear esteemed opponent, thank you for your question. Could you repeat your question again?
Okay, as we know that the induction of C-force, the process of C-force to become a protein, it takes about 90 minutes to 2 hours, and also synaptophysing also from several minutes to hours, okay? And also the induction of neurogenesis, which takes about weeks, about let's say 4 weeks for it to become a mature and functional neuron. And you have demonstrated that short period of stimulation enhances cognitive or memory enhancement. And how do you know that your stimulation enhances memory is through neurogenesis, C-force, and also synaptophysing?
You mean the C-force, the progression of C-force will become a protein. It takes about
90 minutes to 2 hours.
minutes to 2 hours
For it to become a translation protein.
And also the induction of neurogenesis will take about
4 weeks.
4 weeks. How to demonstrate that the short period of the stimulation?
Yeah, how do you know that your memory enhancements is due to neurogenesis?
We in our research, we first used the object location task to use the object location task to find which stimulation parameter is the optimal stimulation parameter, and then we use the BRDU, and after 4 weeks, we find the neurogenesis in the hippocampus, and then after 4 weeks, we perfuse the rest, and I think that will be relationship between the memory enhancement due to the neurogenesis.
Well, your stimulation will be a short period of stimulation. Within the first, let's say, after your stimulation, within the first week, it is still a nesting, it's still an immature and non-functional neuron. Do you agree with me?
Yes.
So, and it takes about 4 weeks for the neuron to become functional, and electrophysiological functional, and also mature neuron. So now my question is, during your first time stimulation, how do you know that this is due to the stimulation effects? Or do you have better other mechanisms that suggest it's a transient change of stimulation?
Actually, in our research, we did the, I agree with you that in our research, we did the acute stimulation, and then we inject the BRDU to see if there's some neurogenesis. And then I think that is a potential mechanism that the neurogenesis has a relationship between the has a relationship with the memory restoration, and I think in the future research we would like to confirm it is it is because of this pathway to restore the memory.
All right. So if that just now you Professor Ayam Blokland asked you about the connection, and you have mentioned that there's no direct input of cholinergic projection to the hippocampus. Then why do you look at the acetylcholine, changes of acetylcholine?
You mean the changes acetylcholine in the NBM-DBS?
Yes. Just now you
there
say
is-
that there's no direct input of cholinergic projection to the hippocampus.
Yes. Dear esteemed opponent, thank you for your question. The hippocampal acetylcholine level were not affected by the NBM-DBS. Since there is no direct hippocampal cholinergic input from the NBM, it has been claimed that the anticholinergic action of scopolamine may more readily impact the hippocampus than other structures. Previously, we have found that the phonics DBS is able to reverse the scopolamine-induced dementia by increasing hippocampal acetylcholine levels. Although it has been shown that the cholinergic neurons in the media septum rather than the NBM regulate the hippocampal circuit. We wanted to investigate the possible of the circuit-wide effect of the stimulation, where NBM-DBS could indirectly modulate the media septum of the hippocampus, and the memory enhancing effects of NBM-DBS might therefore be related to the enhanced cholinergic modulation of the neocortex, and several microdialysis studies of other groups were also able to show that the NBM-DBS applied both continuously and intermittently enhanced the release of acetylcholine in the front parietal cortex.
Do I have time for next question?
No,
All right, so I will end here. Thank you.
Thank you,
Professor.
Dr. Lim.
Thank you, Professor.
The opposition will be continued by Dr. Michielsen. Dr. Michielsen is postdoc at the Department of Neurosurgery at Maastricht University. Dr. Michielsen.
Dear candidate, dear Liu, first of all, I would like to congratulate you with finishing this piece of work. I would also like to extend the congratulations to the team, of course, the promotion team. Reading your thesis, you can really see that you did a lot of work, especially in the lab downstairs with the rodent studies, of course. Took a lot of effort, I believe, to put this all together. But I'm here today actually not to only give you congratulations, but to be critical on the work you did. And would like to zoom into chapter 5, because that's most in line with my research I'm doing. So research on patients, of course, and patients diagnosed with Parkinson's disease. And would like to discuss with you about the methods you used in there. Because in this study, I noted that you actually made use of 10 patients. You included 10 patients. To me, first, it is relatively unclear how these patients were selected. So I'd like to know how you actually recruited these patients. So was there a strategy to actually include them? And then next, I was also wondering about the number of patients used in this study. So you describe in your study setup that you're going to apply this intermittent DBS and continuous DBS. And you decided on using 5 patients per group. So I was also wondering about why did you select only 5 patients per group? And also, would this be enough to actually prove the point here that this cognitive functioning is improving in group A, the intermittent group? So I was wondering about these methods. Do you have enough patients included in the study to actually prove this concept? So could you maybe please try to extend on that?
Dear esteemed opponent, thank you for your question. I think you mean the exclusion or inclusion criteria in my research, right?
Indeed.
Yeah. In our research, all the patients met the diagnostic criteria for the PD patient based on the United Kingdom Parkinson's Disease Society Brain Bank. and all patients had been diagnosed by neurologists in our hospital, specialized in movement disorder, and they must had no other neurological and mental disease, such as
Sorry,
depression.
so these things I know indeed from the thesis, what you wrote down there. But I want to know actually how these patients were assigned to each of the groups. So the intermittent group and the continuous group.
Okay, and for the intermittent group and the continuous group, I divided them two groups by random.
So you flip a coin then each time, or you have somebody independent, or how do you go for the decision to be in group A or B?
For one patient, because there are not a lot of people during 1 month or 2 months, such as one patient will come in this week and another patient will come in next 2 weeks. So I think in my criteria, this person is a group A and the next person is group B. This is a random.
Okay. So it depends on the week, you say?
Yeah.
Okay.
Depends on which patient will come. Next patient will come.
And how confident are you in the findings here? Because it's only a group of 5 patients. You're going to compare with also 5 patients in the other group. How confident are you in the numbers there?
Yeah, I agree with you. And this is still a problem in my research. I think and the sample size is not enough for me. And I think in the future, I would like to expand the sample size, and I will do the research with more PD patients.
That's good to hear that this is going to be extended. Hey, so this is the first setup, of course, to see how it works. And yeah, that's really nice.
Yeah, I think we don't have the sample size, and I write this limitation in my thesis. I think it's really a big problem, and in the future, I would like to expand the sample size. And during that time, I think there will be little bias.
I also have a short question on the methods, because you talk about fusing the CT images with the MR images, and of course it's my expertise as well. And I just want to know about how that actually took place. So what kind of methods did you use to combine the CT image on the one side and the MR image on the other side? Because these are different modalities, of course.
Could you try to give a short answer?
You mean how to combine the CT image and the MRI image?
Indeed, right? Yeah.
I think we use structures, the structures in the around the human head.
Mm-hmm.
And I think we use the titanium alloy structures.
Mm-hmm.
That will be better for the CT image and the MRI image. And we use that to do the surgery with titanium pneumo,
right.
Pneumo alloy.
Yeah. Okay. Yeah, thank you for your answers. I will give the word back to the prolector now.
Thank you very much. The opposition will be continued by Dr. Riemens. Dr. Riemens is postdoc at the Department of Psychiatry and Neuropsychology at Maastricht University. Dr. Riemens.
Yes, thank you, Prorector. So, dear candidate, of course, I also would like to thank you, congratulate you first, as well as your supervisory team on this great milestone and your thesis, which I, of course, also read with great interest. But of course, also there I had some questions, and I specifically want to look back to chapter 4, where you have tested the deep brain stimulation in the nucleus basalis in this animal model of Alzheimer's disease or dementia. And I was actually wondering whether you could speculate on how you see this approach being translated to the clinic. Of course, if we think about patients or Alzheimer patients specifically, of course, we know that these patients are subject to neurodegeneration. Of course, this is something that was not reflected in your model. So I can also imagine then, of course, when you would eventually treat patients with deep brain stimulation, that results might look very different as compared to how you observe that now in your animal model.
Dear esteemed opponent, thank you for your question and thank you for your kind words. For my research consists with these findings. By studying the experimental model of DBS in dementia, we may have found that this treatment schemes is not only suitable for patients with dementia, but also for patients with other central nervous system disease with cognitive impairment. For example, the depression, the obsessive compulsive disorder, and the addiction often suffer from cognitive impairment. Therefore, these findings provide the preliminary evidence to help clarify how DBS may improve cognitive function in dementia and other neurological mental disease.
Okay, yes, thank you. But if you think about Alzheimer patients in the clinic itself, so now you have a model where the nucleus basalis is completely intact. And if we look into the clinic, you might find patients that have been subject to neurodegeneration, especially the later phases of the disease. It could then, of course, easily happen that the nucleus that you're stimulating right now has less neurons present or other cells that play a role in forming the connectivity of the brain. So do you then think that stimulating patients in a very severe state of the disease could be effective in that respect? Do you expect then similar outcomes as compared to what you found now here in your animal work? Or might it be that the effectiveness of the nucleus basalis stimulation is more for earlier phases of the disease?
Dear esteemed opponent, thank you for your question. I think when the patient in the not in the early stage, I think the NBM DBS or the phonics DBS will be better in the progression of the early stage Alzheimer's disease. However, the research is still ongoing during the research time. And if you ask me if the person come to my hospital, I think it's not the right time to do the stimulation surgery. I think we need more researchers in the animal model and even in humans. I think we need more researchers, but now is not the good time to do the surgery.
Okay, so you're saying indeed that we would need to figure out indeed how that then works in the clinic with different patients, and to see whether we can then also improve memory there.
Yes.
My second question that I had relates also to the animal model. Somewhere in your discussions, you referred a couple of times to the fact that the model that you're using is not the most optimal model reflecting the clinical situation. Like we discussed before, there's no neurodegeneration present, for example, in the scopolamine treated animals. So my question actually is if you would have the chance to redo the experiment, how would you change this? So would you use another animal model, or would you add something in addition to the model that you were using in order to more mimic the clinical situation and in order to make it more translatable?
Dear esteemed opponent, thank you for your question. I think it's very nice question. And in my research, we use a scopolamine model. And I think scopolamine is used as a standard drug for inducing cognitive deficiency in healthy humans and animals. In some other researchers, they also used the transgenic Alzheimer's disease rat model. However, the validative of scopolamine as a pharmacological model for cognitive impairments has been discussed in the review. And in particular, we found the beneficial stimulation parameters. I think in the future, we need to employ a transgenic model because it's better to mimic the disease as closely as possible. And we are aware that the model applied in our study does not resemble human AD. For instance, it lacks the AD-specific neuropathology and progressive nature of the disease. Nevertheless, it is an adequate and widely used model to evaluate the potential of experimental therapies on cognitive function. However, for our research, the scopolamine-induced model was a good suit because we only want to investigate which stimulation parameter to use. And I think for the next step, we wanted to see if stimulation also has an effect on the transgenic AD rat model.
Okay, well, thank you. And I'm satisfied with your answers, and I would like to give the word back to the pro-rector.
Thank you, Dr. Riemens. Then we go to our last opponent, Professor Vandenhofer. He's Professor of Epigenetics at Maastricht University. Professor Vandenhofer.
Thank you very much. First of all, compliments and congratulations to the candidates for the thesis and also for the very nice presentation, I have to say. Also compliments to your supervisors. Time is short, so I will be quick. You showed in chapter 4 that the most optimal stimulation paradigm was able to induce long-lasting effects at the level of neuroplasticity. You reached, or you came to that optimal stimulation paradigm by actually applying all paradigms to all animals in a sort of a randomized, balanced fashion. Wouldn't the fact that you see long-lasting effects argue against the repetitive use of DBS in all these paradigms? So wouldn't you expect carryover effects, for example? Alternatively, you could also say, hey, if these mechanisms are influenced by this optimum paradigm, why is then memory not improved also at later time points where you test certain paradigms and certain settings? Because you do know then that in the background, hippocampal plasticity, for example, would be improved. So what is happening here? Do you have an explanation there?
Dear highly esteemed opponent, thank you for your kind words and thank you for your question. And then, yes, we consider carryover effect of the stimulation. During the first trial, the apparatus contains two identical objects at the midline of the iris. After the first exploration period of 3 minutes, the rat is placed back in his home cage, and subsequently, after an interval of 6 minutes, the rat is placed back in the apparatus for the second trial of 3 minutes. And we then did the repeated experiment in the next 24 hours. And in our result, we did not show a pattern of gradual improvement or impairment. So I think it is unlikely that a carryover effect or neuroplasticity occurred over the short time period of stimulation.
Laura asked.
Conversely, yeah.
Sorry, yeah, if you want to shortly finish your answer, go ahead.
Yeah,
Short.
conversely, it's more likely that the changes in performance were related to the acute stimulation. That's my answer. Thank you.
Okay, thank you very much. Huajie Liu, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense, and I request that you and your company await the results of our deliberations and our return.
Thank you.
Luke, can you please transfer us?
Please proceed, Madam Pro-Rector.
And your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Temal is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Huajie Liu, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university shown by the beadle. The laudatio will be done by your co-promoter.
Dear Dr. Liu, dear Huajie, I would like to congratulate you, your family, and your friends with this critical milestone in your life. This is a great achievement. Hora est, it has been done. You're a PhD now, and I have the privilege to say a laudatio. I will do so by telling more or less how you came into our group and how I got to know you as a scientist and a person. So it all started 4 years ago when Yasin suddenly mentioned to us in a lab meeting that a neurosurgeon from China was joining our group. You arrived in Maastricht, and I remember our first meeting quite well, even without 4XD brain stimulation, because it laid the foundation of a very big misunderstanding. I asked you if your name was Liu, and you said yes. And then from that moment on, we all called you Liu, and only at the moment of your first publication, we noticed that Liu is actually your last name. But for us, it was too late to change that, because our nucleus basalis of Meinert already released sufficient amounts of acetylcholine into our neocortex, and the information was already in long-term storage. With respect to the science you performed in Maastricht, you did an excellent job. You're a fast and eager learner. You came to Maastricht to finish the biochemical and immunochemical measurements of one study that was related to magnetothermal DBS. And then, of course, there was chapter 4, your very own animal experiment, with lots of ups and downs, but with a very nice publication as a result. You were always very committed to your projects. You enjoyed presenting your data at international conferences and even won a brand new Red Brain Atlas for your achievement as best poster presenter in Marrakesh. And your commitment was so high that you even took this Red Brain Atlas, and I mean the hard copy, to the desert in Morocco. You know, for some light evening reading under the stars. So during your third year, you intended to go on holidays to China, and that was right before the COVID outbreak in Europe. So I didn't know that that would be the last time for me to see you in person, because you were not able to come back for a long time. You started a job in China, a very prestigious job as a neurosurgeon at Shandong Provincial Hospital. And you were very eager to continue your PhD, and you started a clinical study in Parkinson patients, your chapter 5, which produced very interesting data that we want to follow up on. Liu, it was a great pleasure having you here. We also learned a lot from you with regard to Chinese culture and customs. You're a kind person and you made many friends. And now I would like to close this laudatio by trying to say congratulations in Chinese. Dr. Liu to her. Congratulations.
Thank you, Sarah.
Thank you so much. Dear Dr. Liu, also on behalf of Maastricht University, I would like to congratulate you with the degree you have acquired. Of course, I also like to congratulate your family, friends, and colleagues here and in China. I'd like to congratulate your supervising team with this great achievements. And I'd like to thank all members of the Corona, and especially for our guests from China, Professor Zhang Chen and Dr. Lim, for their time and effort at being part of the assessment committee of the thesis, and here as member of the Corona during this academic session. Dr. Liu, I wish you a lot of success with your further career, and I hope you will continue with the work you've done here and apply the knowledge you have gained during your PhD. And because I think this is a very important topic you've been working on, and I wish you a lot of success with your further work. That's it for this ceremony. With this, I'd like to declare the ceremony for ended.